Alrighty, so we should be ready to go. Okay, so we're continuing on here with post-Mendelian genetics, and so here are a couple of the terms that I didn't get explained so we could, get to the lab and the football game on Monday. Okay, so the next thing we have is called pleiotropic. Some people will pronounce it pleiotropic. I say pleiotropic. But basically, this is when a single gene determines more than one phenotype. Okay, so you have one gene, but it's affecting multiple phenotypes. So, for example, the gene that is responsible for the phenotype of the length of your arms, okay, is also responsible for the lengthening of the legs, because that makes sense. Because you don't have people walking around with really short legs and really long, really short legs and really long arms where they're like dragging their knuckles, okay? Because that wouldn't make sense. Or you don't have people walking around with really long legs and really short arms, kind of like T-Rex. They grow in proportion to each other because there is one gene that is responsible for those two phenotypes. Another example where you have one gene that affects multiple phenotypes is the gene for sickle cell anemia. Okay, so individuals that inherit sickle cell anemia, basically here you have a normal red blood cell. Okay, individuals that inherit two recessive alleles for sickle cell anemia, they're affected. So they're homozygous recessive. All of their red blood cells do this sickling shape, which means that they can't carry enough oxygen, so therefore they're anemic. There's lots of other things that aren't great that affect them as well if they have sickle cell anemia. Now, carriers of sickle cell anemia, individuals that are heterozygous, okay, so they only carry one recessive, they're not affected, but they do have they do produce some sickled cells, red blood cells, but not enough for the full expression of sickle cell anemia. But what has been found is that individuals that are either have sickle cell anemia, full-blown sickle cell anemia, or that are carriers of sickle cell anemia have resistance to malaria. So if you look at this map here, okay, so this is Nigeria, okay, so in these areas that are red, notice that there's lots of mosquitoes there, and then in here it's more desert and arid, so not a lot of mosquito growth. So what a researcher noticed when they were studying sickle cell anemia and they were mapping out the distribution of sickle cell anemia, they noticed a peculiar pattern, that the areas that were highly infected with mosquitoes, okay, and which carry malaria, that's how you get malaria, you have to be bitten by a mosquito carrying the parasite, that in those regions that the number of individuals with sickle cell anemia and carriers of sickle cell anemia for the gene were much higher in proportion than in the areas where there weren't a lot of mosquitoes and not a lot of malaria. And you can see this all throughout the world, that anywhere where there's a lot of mosquitoes, there's going to be, the chance of getting malaria. That individuals in those regions have that sickle cell anemia has a higher incidence in those areas. Pardon me. In those areas. So in this case, the sickle cell gene, okay, affects not only the blood phenotype of the red blood cells, but it's also affecting the phenotype of, are you going to be susceptible or not to, or I should say immune to getting malaria. Because again, carriers and individuals with sickle cell anemia have a carriers have a partial immunity, and those affected are completely immune to malaria because the parasite can't get in the sickle cell shaped. So again, one gene, multiple affecting multiple phenotypes. Okay, then we have what's called polygenic traits. So poly, remember poly means many. So polygenic traits are when one trait is affected by multiple genes. So you need multiple genes working together to result in that trait. And so what I want to kind of give a little explanation here as a visual is think of dice, okay? So dice, right? You have 6 sides, okay? 1, 2, 3, 4, 5, 6. So if you have a pair of dice, and if you were to roll them and map or graph the sum, so for example, if you get a 1 and a 1, that's a sum of 2. So you graph how many times you get each one of the sums. You will see a very interesting pattern. You will see what's called a normal distribution or a bell-shaped curve, where the most sums that you're going to see are going to be, so you can see the ones with the most combinations. Okay, so look at a 7. Okay, you can get a 7 1, 2, 3, 4, 5, 6 different ways you can get a 7, but you can only get a sum of a 2 only one way. You have to roll a 1 and a 1, or a sum of a 12. You can only get that one way. You have to roll a sum of you have to get a 6 and a 6. So when you're rolling dice, just the law of probability, if you roll them 100 times, if there's more opportunities to get a sum like of 7, then it's naturally going to come up more often. But a sum of 2 or 12 is not going to come up very often, because again, you only have one way to get that. Okay, so let's apply that to genetics here. Okay, this is a trait that is affected by multiple genes. So these genes are not discrete. What we mean is, we've been talking about, think of like flavors, you know, chocolate and vanilla, that's it, okay? But just dominant, just recessive, you know, just tall, just short, nothing in between, just purple or white, nothing in between. Well, in characteristics that follow polygenic inheritance, there's no, you know, black or white, okay? But you have all the shades of gray in between, okay? Because the phenotype is continuous. So you've got the two extremes of pure white and pure dark black, but then all in between you have all the variations of the gray in between, okay? So these are things like skin color, height, we think intelligence, and so here, if you were to take a random sample of the population and ask those individuals to line up by height, then you will see this normal distribution here. Okay, very few individuals are super tall and very few individuals are super short. You have total continuous variation of the phenotype. Okay, so think of this as the sum of 2 and this is the sum of 12, and then you have 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, okay, like in the rolls of the dice. When there's more genes involved, like if human height, we don't know exactly how many genes are involved in controlling human height, but let's say that for our example here, there are 6, okay, 6. And then one from mom, one from dad, right? Okay, so the only way to get super short people is you have to get the combination of, let's use the dice, a 1 and a 1, okay? The only way to get super tall people would be to have the combination of a 6 and a 6. But then we have so many people at the average expression of the height, okay, of the phenotype, because this is like the 7. There were 6 combinations that resulted in a sum of 7. So in polygenic traits, the average expression of that characteristic, like human height, you have way more genetic combinations of those multiple genes involved in the phenotype that will come up, resulting in average, but very few in the two extremes. Okay, so that's polygenic. This video here by, it's called Bosman, Paul Anderson. explains polygenic traits really well. If you need more of an explanation, it's, follow this link to YouTube and fast forward it to about 8 minute point 40 second. Gives a great little example of polygenic. Okay, then we have sex-linked traits. Okay, so sex-linked traits. So if you're looking here, I see the number 15, but some individuals that are colorblind, they don't see a number at all. This is a test for colorblindness. Certain characteristics are what we call sex-linked because they're carried on the female X chromosome, and the region of the X chromosome that the Y chromosome doesn't have any physical chromosome there, okay? So we see these more often in males affected than in females. This is a color pattern in cats that is also sex-linked. So what do we mean here? So again, you have your X and your Y, okay? These are what the genotypes would result, the possible genotypes involved in sex-linked inheritance. So a normal female, this is a female who's homozygous dominant. She would have, and you need to represent the sex chromosomes, and then we superscript the gene. So I'm using C's for colorblindness here. Okay, so a normal female would be homozygous dominant. So she carries 2 dominant alleles for colorblindness. A carrier female, okay, she's heterozygous. So she carries a dominant and 1 recessive. So she does not have colorblindness, but she could pass it on. An affected female, which is really rare in sex-linked inheritance. Okay? So an affected female, you can see here that she has to inherit 2 recessives. She has to be homozygous recessive, getting 1 from her mom and 1 from her dad on the X chromosome. Now for males, there's only 2 genotypes and 2 phenotypes. So a normal male, okay, only inherits this from the mom because she's the only one that passes on the X chromosome. And again, this is the portion of the X chromosome that those genes are located, that there's nothing on the Y chromosome there, okay? So for a normal male, a normal vision male, not colorblind, he would inherit a dominant from his mother. Doesn't inherit anything on the Y from the father. So an affected male only has to inherit 1 recessive, and they get it from their mother. Okay, it's passed on from their mother. So here's an example of hemophilia, which is bleeder's disease. And so here you have the male, okay, so normal, non-affected male, okay. And then here you see that the mother is a carrier, okay, so there's the gene for hemophilia. So when they have kids, okay, we represent, the girls that they could possibly have and the boys that they could possibly have each time that they have a conception occur. So the dad could pass on, here we have the X chromosome to his daughter. So the dad is going to pass on the dominant to all of his daughters because that's all he has to pass on to his daughters. The mother is going to pass on, she could pass on either the dominant on her X chromosome, so that would be a normal non-carrier female, and then here's her other daughter right here, and you could see that the mother passed on a recessive gene for hemophilia, so this daughter is just a carrier. She doesn't have hemophilia because, again, for females, she would have to inherit 2. But the boys, okay, what is the male going to pass on to the boys? Just the Y chromosome. So it's mom that's going to pass on either the dominant to her son for a normal non-affected son, or she would pass on the recessive to her son. So in sex-linked conditions like hemophilia, Duchenne's muscular dystrophy, and colorblindness, an affected male always inherits it from his mother. So his mother has to be at least a carrier. Now, how can you get affected female? An affected female, okay, a girl having it, she would have to inherit, here's the affected female, she would have had to inherit one of these from her mom, so her mom must have been at least a carrier, and her dad had to be affected, okay? So she has to get it from an affected female from both parents, okay? Alrighty. So here's a practice problem. Both a mother and a father of a colorblind male, okay, appear to be normal. From whom did the son inherit the allele for colorblindness? Okay. Well, so the son is going to be XY, right? He's going to be XY. And so he inherits the Y from his dad. And since the dad doesn't have color blindness, the dad passed on the Y chromosome to him. So where did that affected gene have to come from? It had to come from mom. And since mom doesn't have color blindness, mom is a carrier. Okay? So the genotypes of the mother would be a carrier female. Okay? The father was a normal male, and the son is an affected male. Okay? Alrighty. Now, there are some characteristics that the genes are actually turned on by the environment. So it's genetic, but it doesn't get turned on or off unless the environment has an effect on that. So if you look at Himalayan rabbits, okay, so here are the extremities, here, where the color pattern is. And that's where they're losing body heat. Okay, they lose body heat out of the extremities, and so in that case, okay, you see that the pigmentation gene is turned on when it's colder. Okay, so when it's colder, the temperature is affecting that pigmentation gene being turned on. Okay, so you could take an ice pack and you could put it on the bunny for a period of time, and what would happen is the temperature would turn on that pigmentation gene in those hair follicles, and you would see that the color pattern, the pigmentation would be produced. Okay. All right, so that is going to be it for what we didn't finish on Monday. Okay. All right, we'll see you next week.